Speciation, the formation of new and distinct species, occurs through various modes, primarily categorized by the geographical context of the reproductive isolation driving the divergence. 1. Allopatric speciation. 1.1 Definition. This is the most common mode, occurring when a population is geographically separated into two or more subpopulations, preventing gene flow between them. This separation might be caused by factors like mountain formation, river changes, or continental drift. Over time, these isolated populations accumulate genetic differences due to different selective pressures, genetic drift, and mutations. 1.2 Types of allopatric speciation. There are two subtypes, vicariance and dispersal. Vicariance involves a geographic barrier splitting a previously continuous population. Dispersal occurs when a small group colonizes a new area, geographically isolated from the parent population. 2. Parapatric speciation. 2.1 Definition. This mode involves the divergence of populations that are adjacent to each other, with limited gene flow between them. Accline, a gradual change in allele frequencies across a geographical region, often characterizes this. This limited gene flow might be due to a gradual environmental change across the habitat, leading to different selective pressures in adjacent areas. 2.2 Mechanisms. Hybrid zones between diverging populations can form, with varying degrees of gene flow and selection against hybrids, contributing to reproductive isolation. 3. Sympatric speciation. 3.1 Definition. This mode is rarer and involves the formation of new species within the same geographical area, without any physical barrier to gene flow. 3.2 Mechanisms. Several mechanisms can lead to sympatric speciation. These include sexual selection, non-random mating based on mate preference, polyploidy, a sudden increase in chromosome number, mostly in plants, and disruptive selection favoring extreme phenotypes over intermediate ones. 4. Parapatric speciation. 4.1 Definition. A special case of allopatric speciation, parapatric speciation involves the colonization of a new habitat by a small group from the parent population. The founder effect, where the new population's gene pool is a non-representative sample of the original population, plays a significant role. 4.2 Genetic drift. Genetic drift, random fluctuations in allele frequencies, is a significant factor in parapatric speciation because of the small initial population size. 5. Artificial speciation. 5.1 Definition. Human intervention can also drive speciation. This can be through selective breeding of domesticated animals and plants, or by directly separating populations to prevent gene flow. In conclusion, speciation is a complex process driven by various factors, including geographic isolation, genetic drift, natural selection, and sexual selection. The modes of speciation, allopatric, parapatric, sympatric, and parapatric, represent different ways in which reproductive isolation arises and leads to the formation of new species. Understanding these modes is crucial for comprehending biodiversity and evolutionary patterns.